Great. Thank you for that fantastic talk, Dr. de Botton. We're going to take your poll again, and everyone, in case you didn't notice the last time, this is actually, there's no right answer. It's a, what is your opinion on how you would take care of this patient? So again, it's a 72-year-old man with newly diagnosed MDS with excess BLAST2, who has a good performance status and a P53 mutation, but with a normal karyotype. And this has two parts to it. The first is what you would do for initial treatment, as well as what you would do to consolidate if the patient gets a complete remission. So the first question is, which treatment would you recommend for the initial treatment? 7 and 3 plus gemtuzumab, ozigamycin, CPX-351, 10 days of decitabine, venetoclax plus azacitidine. So predominantly people would have chosen venetoclax plus azacitidine. And the second part of the poll is coming up. So if this patient now achieves a complete remission, would you recommend allogeneic stem cell transplantation for this patient? Yes, he's in good shape. Yes, the prognosis is very poor without allo transplant. No, the outcome of P53 with MDS with excess BLAST2 is poor even with allo transplant. No, he is too old, or only in the case of P53 mutation clearance. Okay, great. So mix of opinions here, evenly spread, except for the no, he is too old. Most people would not choose that one. All right, so Dr. de Botton, maybe you would give us your opinion about how would you induce that patient, and would you how would you consolidate that patient if they received a complete remission?
Well, thank you. What we've seen in the presentation is that when you do have only one allele mutation, you may treat this patient with intensive treatment. A 777 would be the option I would prefer, followed by stem cell transplant, because the efficacy with only one mutation is roughly exactly the same as in case of wild type P53. Therefore, I would follow something very conventional, intensive treatment followed by stem cell transplant.
All right.
Jane, can I make a question?
Okay. I would love it.
So, Stephane, I'm not familiar with the mechanism of action of APR-246. You correctly mentioned the paper by Elsa Bernard in Nature Medicine, showing that patients with biallelic TP53 mutations have regularly an unfavorable outcome. They do not respond well to transplantation, to azacitidine, and so on. What about using APR-246 in these patients? I mean, I only read that the drug reactivates TP53, normal TP53, but if these patients have biallelic mutations, are they still likely to respond to treatment or not?
They are very likely to respond, because if they do have mutations, there's a missense mutation in DNA binding domain. And with APR-246, the MQ, which is the active drug, will fix the molecule in, will fix P53 in a normal conformation and reactivates P53 functions. So it is obvious in these trials that they do have multiple alterations those patients had abnormal karyotype, complex karyotype, and p53 mutations. So they have double inactivation of p53, either by loss of p53 or by mutations. And what we've seen in this trial is that you may increase the proportion of responders, which is really very important. And the drug seems to be very effective. Furthermore, you can follow the decrease of the VAF, suggesting of the activity of the drug in that situation. So we have different things that suggest that the drug is very effective, even in those poor risk patients.
So the answer is that you did treat patients with biallelic mutations and showed a response to APR-246.
Yes, I think that the selected population is the worst population in those trials. It's obvious because they have complex karyotype. And as you noticed, more than 90% had biallelic alterations in that case. So this is the worst population that you may select in that case. That would be also interesting to note if we do have better answer in case of only one allelic alteration, one mutation with the drug. It's not known yet because most of these patients, the vast majority, do have double inactivation of p53, either by missense mutation or loss.
It's very important. Thank you.
Yeah, my question is sort of on the similar line. So following up on that, if you obviously if you have p53 around, even though it's mutant, this drug can work against that p53. But if you have biallelic loss, so you have del 17 on both alleles, for example, and you don't have p53 around, that's probably a small subset of these cases. But I would wonder if this drug would be able to work in that context.
So if you retain one p53 gene functional, the outcome is equivalent to both functional genes. This is what we've learned from the study. And with the drug, you may change the conformation of the protein, even if there is only one mutation and the other gene is lost. For instance, in case of 70p. So the importance is to retain one functional gene, and the use of the drug may change the functionality of the mutant gene. So that's very important to notice.
Great. Dr. Archana Shetty is asking, when do you expect the results from the phase 3 1533 trial of APR-246?
Probably early next year, because the accrual has been completed in June or July, as far as I remember. So we should have these results sooner or later. That's very important.
And Dr. Elsa Bernard is asking, is APR-246, are those trials open to patients who have truncated p53 mutations, which are 20% of mutations? What is the response in this patient category? So sort of along the lines of what I was asking, if you have a truncated version, are those patients going on and do they respond?
If you don't have p53, which is a rather rare situation, the effect of the drug has not been precisely studied until now, but probably no effect. I don't know precisely. But if you retain one mutated allele, which is the vast majority of these patients, you do have the very high response rate, above 75% of overall response rate.
Great. And can you comment a little bit more? Obviously, we're really excited about a drug for this really refractory subgroup. So can you comment more on the neurologic toxicity that is being observed with APR-246?
There was a high percentage of neurotox, but these were grade 1, 2, ataxia, dizziness, and it was manageable because these side effects fully disappeared after drug discontinuation. So this is quite easy to manage.
Okay, great. And do you think that we will get to a molecularly based trial where we can randomize patients at diagnosis to different arms of all of these different targeted agents, or are they too infrequent overall to make something like that feasible for this disease?
We can envisage lots of things with such a type of very well tolerated drug. Again, that's really very encouraging. If you increase the response rate, which is the goal of the randomized phase 3, we will have the results probably by the end of the year or early next year. Again, it will provide us a strong rationale to use the drug frontline. It's rather safe and easy to manage. Side effects are not too strong, and we do not observe increased cytopenias with the combination. So we can envisage lots of combination with intensive treatments, but also following stem cell transplant, which is another important question.
All right. And can you comment on APR-246 as maintenance after transplant?
So what we've seen in that huge paper, the outcome is rather poor. It's better than nothing, but it's poor even in case of stem cell transplant. And we can envisage maintenance maybe in combination with low dose RSE, and yet that we may have oral RSE. The combination could be great after stem cell transplant to delay relapse or to avoid relapses, and that could be something that could be manageable in the post-transplant setting. That's a great question.
Okay, great. So I think thank you very much for the answers to those pressing questions and for your fantastic talk on these targeted agents.
Thank you.
So now I will offer very brief conclusions and perspectives, and then we'll get you guys all on to the rest of your ASH seminars. So I have no relevant disclosures. So first and foremost, thank you to all of you for attending. We had over 400 people attend today from wherever you are in the world, and you all contributed to excellent questions and learning more about the current state of treatment and molecular diagnostics in MDS. And one logistical thing, just watch out for a feedback survey that you're going to get an email from us. This will help you to get your CME credits as well. Thank you to all of our excellent speakers for their time today and their excellent presentations. And thank you to the MDS Foundation board and staff. I mean, during this year has been absolutely crazy, and obviously this has shown us ever more that having an international group of MDS expert caregivers and clinicians and researchers all working together is critically important. And so thank you, and thank you for organizing this fantastic seminar series and adapting it to our current situation. And especially to patients, caregivers, clinicians, and researchers worldwide working together to try to optimize the care of our patients, quickly adapting to this situation in real time as things change day to day. So I wanted to just take a moment of silence. This is a map of all of the deaths that have been experienced worldwide because of COVID-19, just as of November 27th. So in memory of people who have lost patients or family members, colleagues, and others. So I think that today we've seen that there's definitely lots of resiliency in our communities. Global collaboration has been absolutely key. And we can tolerate more change than we'd like to admit. There obviously have been major challenges, and Dr. DeZern's talk really showed that nicely. How have we been working to keep patients and providers safe during this pandemic, implementing new models of care and doing that really quickly. So we went from all in-person visit to all telephone to all video within a span of 1 week's time. So those feats are definitely very, very challenging for everybody, but also open the door to new ways to care for these patients. And we've had lack of key needs like blood supply and clinical trials and research. But the major highlights have really been that we've learned these new efficient ways to deliver care. Hopefully we'll all have shot and blood drive-up windows as a part of this. and the key global collaboration. It's been great to watch academia and pharma accelerating the timelines for testing for this virus and for vaccination, and hopefully we'll all be getting vaccinated soon. So our second theme today from Dr. Rafael Bejar was expanding the utility of genomic profiling in MDS. And this was also the theme, one of the themes of Dr. de Botton's talk because of the new targeted agents. So genomics are helping with diagnosis between benign cytopenias and MDS and differentiating between myeloid disorders. Also for prognosis, the single versus combinations of these mutations, the variant allele frequencies. One perfect example is monoallelic versus the biallelic TP53 mutations. And now the promising use of these molecular profiles for therapeutic decisions, which will definitely facilitate us having more and more demand for getting these tests to our patients. So the US already, the National Comprehensive Care Network now highly recommends doing genomic testing, and we are anxiously awaiting the International Working Group's IPSS prognostic score that will now incorporate these molecular profiles. And so our MDS community really needs to help advocate for getting access to this testing for our patients. We're going to have to share data and contribute to these consensus efforts like the ASH Precision Medicine Initiative that Dr. Bejar mentioned to help us make sense of all the rare variants that we're going to see in these patients and make sure that we can find the ones that are actually going to make a difference for our patients. And the last theme is that there are promising new therapeutic options on the horizon, which is fantastic news for all of us and our patients most importantly. So luspatercept has been approved in 2020 and has been useful in this COVID era, as Dr. Dezern mentioned. Dr. de Botton very nicely showed us how single agent and or combinations with HMAs for venetoclax, APR-246, and the IDH inhibitors. And as Dr. Gill said very nicely, we're at the end of the beginning of learning how novel CAR T-cell strategies will be incorporated into MDS care. And so this horizon is a little farther off than the others, but we all look forward to figuring out how to incorporate these into our MDS care practices and having more options than just HMAs and transplant for our patients. So I hope all of you hang in there. This is a trying time. If you read Dr. Steensma's, we all have type 4 fatigue syndrome with a much fancier name than I can remember. But there is hope. Already we're talking about getting vaccinations for people on the front line in the next couple of weeks. And so I hope that gets to all of you out there very soon. Definitely stay connected. I hope everyone has a very productive ASH meeting, and you can always look at mdsfoundation.org for help for your patients and to find our IPSS prognostic scoring calculators and other resources. And we really hope to see everybody in person at the 16th International MDS International Foundation Congress in September. So with that, I'd like to say thank you, and we'll end our session today.